Hello, folks, and welcome to part two notes for our unit on gene expression regulation. Here we're going to cover transcription, which goes along with 6.3 in the AP Bio unit guide and 6.4. And you can see the page numbers in our book that go along with that in chapter 17, so you can go there for some extra information. So, quick overview. Just in general, information content of genes is a specific sequence of nucleotides, A, T, TTG, C, T, G, C, A. That is the order. That is basically the instructions for life. It tells you the order in which to put amino acids together and do some various other things so that you can make the enzymes and the proteins that do everything else in your body. So your DNA is inherited and will lead to specific traits dictated by the synthesis of specific proteins. We covered kind of all that in genetics in the last unit. And proteins are those links between the genotype, your sequence of A's and G's and T's and C's, and your phenotype, your hair color, eye color, enzymes in your blood, what have you. So gene expression, the process by which DNA directs protein synthesis, includes two stages, transcription and translation. Now, in biology, we covered the basics of these, and there's really nothing new as far as the basics. So if you're having trouble with any of this, you might want to go back and take a look at some of the stuff I posted from the regular biology class to give you a good overview of this. We're just going to be adding some detail in here as well. A little bit of review and then some extra detail. This stuff here, as always, guys, when I have it grayed out and italicized, it's interesting stuff. It might give you some context for some things, but I don't think it's going to appear on the AP Bio exam. It's not something you need to memorize. It's more the thought process behind it and some of the discovery kind of stuff. So how do we figure out this whole DNA, gene, enzyme, protein connection? Well, George Beadle and Edward Tatum exposed the bread mold they had to X-rays, and they basically mutated it. They mutated it such that they were unable to survive on different media that were lacking different things. So using crosses, they and their co-workers identified 3 classes of arginine deficient mutants, so ones that don't have arginine, each lacking a different enzyme necessary for the synthesis of arginine. So there's a number of enzymes that need to be there to go from the precursor arginine, makes a change, you get a mid place, you make another change with another enzyme, you get a little bit further along, you make another change, and you got arginine. It's kind of like if you had to make a meal where first you had to cut the stuff with a knife, that was one enzyme, then you had to mix the stuff in a mixer, that'd be another enzyme, and then you put it in the oven and it baked it, and that got you your last piece, your final product. If you're missing any one of those, the knife, the mixer, or the oven, you wouldn't be able to finish the process. So from this, they developed a one gene, one enzyme hypothesis, which states that each gene dictates the production of a specific enzyme. It was a start, but it wasn't the end point. Here we can see a diagram of what they did. You can see the precursor for arginine, the midpoints, like here you cut it, bang, you wind up with your cut up pieces. You mix it, bang, you wind up with your mixed stuff. Yeah, bake it in the oven, bang, you got your final thing. Here's the wild type, the different mutants here, and what stuff they can grow in and can't grow in. And down here we can see where the process stopped. And this is what led them to their realization. I'll give you time. You can look at this later if you want to. However, some proteins aren't enzymes, so researchers later revised the hypothesis. One gene equals one protein. And many proteins are composed of several polypeptides, each of which has its own gene. So each polypeptide has its own gene. Like hemoglobin's got a couple different parts, different polypeptides that come together to make it. That's the quaternary structure. So now we restate the hypothesis as one gene, one polypeptide hypothesis. But in general, it's common to refer to gene products as proteins rather than polypeptides. So basic principles. So some of the basic things here, guys, just review RNA. Ribonucleic acid is the bridge between genes and proteins. We got to go from the genes stored in our nucleus into the proteins out in the cytoplasm. RNA is what carries that information out there to build the proteins. Transcription is the synthesis or making of RNA using information in our DNA. We're making a copy of that chunk of DNA, that gene for that polypeptide. Transcription produces, that copy is called messenger RNA or mRNA. The messenger RNA then goes out to the ribosome, which is where translation happens. And translation is where we actually synthesize or make the polypeptide. We put the amino acids together in the right order based on the information that was on that messenger RNA. So that's kind of the overview, big picture. In prokaryotes, bacteria, translation of mRNA can begin before transcription is finished, so it can happen simultaneously. In eukaryotes, it is separated because we do have the DNA stored in the nucleus. We have to finish transcription, then send it out to the ribosome, out in the cytoplasm, and then we can do translation. So there's two separate events in us. And another interesting thing, in prokaryotes, the mRNA that's made from the DNA is it. In eukaryotes, though, the RNA transcripts, the things we make from the DNA, aren't the final mRNA yet. They have to be modified through RNA processing before they get to the final stage that can then be transcribed at the ribosome. And we'll look at that a little more closely in a little bit. So here are just some visual representations of it. We can see a cell, a nucleus, so this is a eukaryote. DNA, we transcribe our pre-mRNA, or RNA transcript as it's sometimes called. Then we process it, do a few things here, add some stuff onto the ends that make it readable. At the ribosome, out here in the cytoplasm, where we do translation and make our polypeptide, our protein chain of amino acids. And here you can see with a prokaryote over here, we can just go bang, bang. We don't have that RNA processing step in there. So like I said before, the primary transcript is the initial RNA transcript from any gene before we process it. And just want to take note of something called the central dogma. I haven't found this vocab term in AP Bio, but just in case, it is the concept that cells are governed by a cellular chain of command. Basically, we go from information in the DNA, convert that to the information in the RNA, which then gets converted into the final product, the protein, which does everything else in the cell. So how are the instructions for assembling amino acids into proteins encoded into the DNA? Okay. There are, if you'll recall, 20 different amino acids, but there are only 4 nucleotide bases in DNA. So how many nucleotides correspond to amino acid? Well, if it's just one, we only got 4 bases, so we can only code for 4 amino acids. If it was 2 base longs, like AT coded for arginine and AC coded for guanine, I think we could only get like 16, so we're still short. So we have to go to 3-letter words called codons or triplets of nucleotides in order to code for all of the different amino acids that we have. So the flow of information from gene to protein is based on that triplet code. Non-overlapping 3 nucleotide words, so to speak, just like our alphabet's got, what, 24, 26 letters in it, and we can rearrange those letters in different ways to make hundreds of thousands of different words. Same thing here with these codons. The words of a gene are transcribed into complementary, non-overlapping 3 nucleotide words of mRNA. So we go from the DNA just through base pair matching like we did with DNA, C with G and A with U. If it's an A on the DNA, it'll be a U on the RNA. If it's a T on the DNA, it'll be an A on the RNA. We will transcribe them into mRNA, and then that transcribed mRNA will be translated into amino acid language, from that nucleotide language to amino acid language at the ribosome, and we'll make our polypeptide. So here, just a visual of it. Here's our DNA, 3' to 5' here. We make our mRNA from this instance. We're matching it up here, right? A with U, G with C, C with G. So we're going 3' to 5'. Every 3 letters is another word. There's no spaces or anything in them like in a sentence. We just know 3, 3, 3, 3 to read it. This 3-letter code translates into tryptophan. This 3-letter code transcribes into phenylalanine, glycine, serine, and so on. By the way, you don't have to remember the codes for each of the different amino acids, and you don't have to remember the names of all the different amino acids. Okay. So during transcription, one of the two DNA strands, called the template strand or the sense strand, provides a template for ordering the sequence of complementary nucleotides in an RNA transcript. The template strand is always the same strand for every given gene. You get two strands of DNA, right? One on the left, one on the right. It's not that just one side is the template or sense strand and the other one is the non-sense strand or the kind of the backup copy. It can switch. It can be the gene could be up here on this strand and another gene can be down here on this strand, but it's always on the same strand. The gene for eye color is always on this strand, never on that strand, no matter which human you're looking at. So during translation, the mRNA base triplets called codons are read in 5' to 3' direction. Okay, so that's the direction 5 to 3. And each codon specifies an amino acid, 120, to be placed at the corresponding position along the polypeptide. Now, with three-letter words, it gives us 64 possible codons. 61 code for various amino acids. 3 of the codons are stop signals to end translation. Okay. And the genetic code is redundant. We got 61 codes for amino acids, but only 20 amino acids. That means we can have multiple codes that will code for one amino acid, just like we can have multiple words that mean dog, right? Dog, perro, canine, right? Different variations, but when you say those, you all get the same picture in your head. Okay. But still all a dog. Codons must be read in the correct reading frame. In other words, these 3, then these 3, then these 3. You can't read, these 3. It won't get you. It won't be right. So it's got to be the first 3, then it reads the next 3, then the next 3. You guys may recall this from bio, mRNA codon charts. I prefer the circular ones myself, but you read them from the inside out. So if the first letter in the codon is a G, and then you move out, and if the second letter is a C, and we move out, it really doesn't matter what the third letter is because GACU all code for GC whatever, codes for alanine. Whereas some of them only have a single one, like UGA, that's a stop codon. That codes for ending the production of that protein. That's going to be at the end of your mRNA. Here are the 3 stop codons. Over here, you can see the highlighted one of the green start codon. Your mRNA molecule starts coding proteins always here with AUG. And this one here, we can also see the different combinations that give us a different 20 amino acids. So the genetic code is nearly universal on planet Earth. It's shared by the simplest bacteria to the most complex animals. Interesting thing is we can transcribe, and genes can be transcribed and translated after being transplanted from one species to another. And that's the whole basis for a lot of our biotechnology and transgenic organisms. Here we can see a tobacco plant with a firefly gene in it, or a pig with a jellyfish gene in it. Okay? The mechanics of transcribing and translating a DNA code into proteins into the cause of a particular phenotype are the same amongst pretty much all organisms on planet Earth, which makes it easy. We just have to take the code from one animal, we can put that code in another, and it can often express it. And that'll do it for the overview. I'm going to stop it here. You can pick up these notes with part 2B, which will be 6.3, transcription, delivering the blueprints.